All right, good evening, everybody, and welcome to tonight's Broad Discovery Series talk. My name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad Institute, and it's a thrill to see all of you in the room with us this evening, especially on such a chilly night. Thank you for coming out and joining us. Tonight, it is my pleasure to bring to the stage Heidi Greulich and Matthew Meyerson. Heidi is an Institute scientist and a senior group leader in our cancer program, while Matthew is an Institute member in our cancer program, the Charles A. Dana Chair in Human Cancer Genetics at Dana-Farber Cancer Institute, and a professor of genetics and medicine at Harvard Medical School. Heidi and Matthew were the Broad leaders behind the development of sevabertinib, a new targeted treatment for lung cancer and the first FDA-approved drug to be developed based on Broad Institute science. They were there through the drug's entire 20-year development history, and they're going to talk about that with us tonight. If you have any questions for us after Matthew and Heidi's talk, and after we have a little bit of a fireside chat, you can flag down mic runners. And if you're watching on YouTube, you can also put any questions you might have into YouTube chat. I'll try to work you into the conversation. If you're a Brody, or if you're a scientist with a deep technical question, I'm going to ask you to hold your questions until the reception after this evening's talk so that members of the community can have a chance to get their questions answered. And speaking of the reception, after we're all done tonight, everybody here in the room is invited to join us for a reception in the Broad Discovery Series or Center right out here outside the auditorium. And with that, I will turn it over to the two of you.
All right, good evening. I'm Heidi Greulich.
And I'm Matthew Meyerson. Thank you all for being here tonight.
And we're excited to tell you about the discovery of a new lung cancer therapy called sevabertinib. So let's start with the most basic question. What exactly is cancer? So cancer is a disease of uncontrolled cell division. It's caused by an increase in function of a growth-promoting oncogene or decreased function of a growth-stopping tumor suppressor gene. So we can think of the cell as analogously to a car, which is normally under good regulation by pressing the gas pedal or the brake as needed. However, activation of an oncogene is like supplying the car with afterburners that propel the car forward whether or not the gas pedal is whether the gas pedal is pushed. On the flip side, inhibition of a tumor suppressor gene is like removing the brakes from a car. Now, this may not be immediately apparent upon standard forward motion, but you can imagine there are circumstances upon which this becomes very consequential.
And oncogenes in particular are activated by different types of genomic alteration. These include mutations, as shown on the left of this slide, which is a change in a single base of DNA, which can turn an inactive gene into an active gene. Then include amplification, where you get extra copies of a gene, and you make more of the gene and that corresponding protein, again, fueling its activation. And finally, they can include breakage and rejoining of chromosomes and genes, which creates a fusion gene, which creates a fusion protein, which now also is inappropriately active, where it ordinarily should be inactive. And so each of these changes drive a cancer, but each of these changes also provide a vulnerability for a cancer.
And so what causes these changes in the DNA that activate cancer genes? So this is data from a study of British cancer patients showing that about 5% of cancer genes are inherited from either the mother or the father. And examples of this include P53 mutations in Li-Fraumeni syndrome or RB mutations in retinoblastoma. Also mutations of the BRCA breast cancer gene that can be inherited are included in this bucket. Second, we have about 29% of cancer gene alterations are due to environmental exposures. And this can include smoking, it can include ultraviolet light exposure, and it includes infection with some cancer-promoting viruses such as papillomavirus. However, by far the biggest bucket is the blue part of this pie that shows that most of the changes that activate cancer genes are due to chance mistakes in DNA replication. In other words, plain bad luck. Now, probably most of you know that lung cancer is largely associated with smoking. However, the mutations we're going to talk about today occur primarily in non-smoking patients and therefore land in the blue bucket, chance mistakes in DNA replication. So Lung cancer is the leading cause of cancer death in the US and worldwide in both men and women. So thankfully, mortality rates for lung cancer are dropping, likely due to a combination of decreased smoking and improved targeted therapies. The 3 types of FDA-approved therapies for lung cancer include old-fashioned chemotherapy, in which a molecule is administered to the patient that preferentially kills the rapidly dividing tumor cells, and less so for the normal cells in the body. It includes immune checkpoint inhibition, which reactivates the immune system to recognize and destroy the tumor. And finally, it includes targeted therapy, which interferes with the function of a usually mutated form of a protein upon which the tumor cell depends for survival.
If we go back to the early days of our project 25 years ago, basically no lung cancer treatment was any better than any other. This is a study of 4 different chemotherapeutic regimens for lung cancer, published in the New England Journal of Medicine, probably the leading journal of clinical research, in 2002. And what you can see here is survival on the vertical axis and time on the horizontal axis. And the median survival with 4 different regimens of chemotherapy was around 8 months. So really, to address this problem and to solve this problem, we began to study lung cancer, and we did so using really new methods that had been developed in significant part right here at the Broad Institute and its precursor, the Whitehead Institute, MIT Center for Genome Research. And this is the analysis of the human genome, as shown here from the International Human Genome Sequencing Consortium, led right here. And this was a collaboration between Bill Sellers, whose picture at the time is shown at the top, and me, whose picture at the time it was shown at the bottom. You know, I hate it when people laugh if I say that. You know, it's just one of those things. But and Bill and I actually were going to a scientific meeting in Switzerland, and we sat on the plane together, and we had a long time to talk about the genome and the advances in cancer therapy. And at that time, not only had the human genome just been sequenced, but in addition, there was a new drug called imatinib or Gleevec for the treatment of chronic myeloid leukemia, which showed remarkable effectiveness for one class of cancer mutations, shown in a couple slides back, the gene fusion that's driving that disease. And so this gave us the idea, you know, if we study more protein tyrosine kinase genes, maybe we could find more mutations that would cause cancer and be targets for therapy. And we really teamed up with a number of colleagues here at the Broad Institute, including Eric Lander, who was the director then, David Altschuler, Stacy Gabriel, who runs the platforms here, and Todd Golub, who's now the Institute director, in initiating this project. And on the next slide, what you can see is we found actually mutations in the epidermal growth factor receptor, or EGFR gene, mutated in non-small cell lung cancer. And what you can see on the left-hand side here is a crystal structure of the EGFR protein, showing what's the protein kinase domain, the domain that actually does the active part of it. And you can see mutations in different parts of this domain, all driving lung cancer. And they're in different spots, and they have different behaviors. One mystery about the EGFR mutations, really, which is not yet resolved, is that these mutations occur in about 1 in 6 patients to 1 in 10 patients with Western ancestry, but about half of patients with East Asian ancestry. And so we believe there's some kind of inherited risk factor that modulates the risk of getting these mutations. And these are somatic mutations. They arise in the body. They're not inherited. They arise in the cancer itself. But we don't know why there's this risk. The other thing I mentioned, that there are mutations in different parts of the protein. And many of these were susceptible to drugs that were developed early on. And I'll say more about that on the next slide. But there's one exception, too, that we'll hear about more, the exon 20 and frame insertions. On the next slide, a little bit about the response. So there were already EGFR inhibitors because people understood that the epidermal growth factor receptor, EGFR, was important for cell growth and division. And so people postulated that it would be important for cancer. And these inhibitors had been used in lung cancer treatment. And what you can see, this is from our colleague Bruce Johnson, the left chest x-ray shows a lung that's filled with cancer. And on the right chest x-ray, you can see within 3 months of treatment with gefitinib, this cancer had disappeared. And our colleague from Vanderbilt and Ohio State, David Carbone, said, we see a complete Lazarus type of response that's never been seen before. But it was mystery why this was. But we knew, I mentioned that these patients, the mutations are more common in patients from East Asia. We knew the patients who benefited from gefitinib were patients with one type of lung cancer, lung adenocarcinoma. They were more frequently Japanese than patients from other countries, more frequently women than men. And as Heidi mentioned earlier, this is a disease that arises more frequently in non-smokers than in smokers. Although I should mention, tobacco smoking is not protective against this form of lung cancer. So the patients who benefited from gefitinib were the same patients in whom we found EGFR mutations. And when working with our colleagues Pasi Jane and Bruce Johnson, we went back and looked for patients who had taken gefitinib and benefited or failed to benefit. The ones who benefited, their cancers had EGFR mutations. Excuse me. And the ones who failed to benefit, their cancers did not have EGFR mutations. So we knew not only is this a new gene that's driving lung cancer, but it's associated with this mystery of response to a drug that was effective for some patients.
However, we realized pretty early on that these early generation EGFR inhibitors would have no benefit for lung cancer patients with EGFR exon 20 insertions. So the experiment on the left, you can see colonies of cancerous cells engineered to express various forms of mutant EGFR growing in soft agar. Importantly, the cells are treated with the first generation inhibitor erlotinib, which behaves much like gefitinib that Matthew was just mentioning, in increasing concentrations from left to right. You can see that two of the mutations, in the exon 19 deletions and the exon 21 point mutation, respond fairly well to erlotinib. And you can see the growth of the cancerous colonies being inhibited by fairly low concentrations of erlotinib. Importantly, the concentration of erlotinib that's required to inhibit the growth and proliferation and survival of these cells carrying the exon 19 deletion and the exon 21 mutation is lower than the concentration of erlotinib required to inhibit wild-type EGFR. And this is important because inhibition of wild-type EGFR in normal cells in the body is one large source of toxicity for cancer patients treated with EGFR inhibitors. On the other hand, you can see in the bottom row that the exon 20 insertions are not well inhibited by erlotinib, and these colonies of cancerous cells continue to grow up until the concentrations of erlotinib that also inhibit wild-type EGFR. Therefore, there's no therapeutic window for this inhibitor for the exon 20 insertions. On the right, you can see patient data. These are lung cancer patients treated with early generation EGFR inhibitors. And you can see that the lung cancer patients with the EGFR exon 20 insertions, shown in red, do particularly poorly when treated with these inhibitors. On the other hand, patients with the exon 19 deletion and exon 21 mutation, together known as the classical activating mutations, shown in black, have much better survival. And in fact, this survival curve is even better today due to the development of further improved inhibitors. So it's data like you see here, experimental results like you see here, that inspired us to commit to developing a treatment for cancer patients with EGFR exon 20 insertions.
And we were very fortunate after looking for ways to advance this research that we met together with some scientists from Bayer Oncology in a cancer research conference in India. And based on this, we started to plan and came to an agreement for a collaboration. And our first project was trying to solve exactly this problem of the EGFR exon 20 insertion mutations. We started in 2013 in this collaboration, and it took 12 years of working together to actually bring the drug sevabertinib to patients. And now we're going to tell that story. We've really had a wonderful collaboration with Bayer. And it's included joint project teams, joint governance, and joint ownership, which has really brought us together in a really seamless partnership and a very open dialogue and exchange of ideas, because we really bring very different expertises to bear in this effort. But we've had exchange of our scientists. Heidi spent a summer in Berlin. We've had their scientists come here. We have joint scientific workshops, and we get together often in joint team meetings and governance meetings, really helping us drive the project forward.
Okay, so as much as can be generalized, here is the path to discovery of new targeted therapies. First, you need a validated target. this is a protein, usually a mutant protein, upon which the tumor cell depends for survival. So for us, this was the EGFR exon 20 insertion. Then one does a high throughput screen, and this involves identifying the few molecules out of a collection of millions of molecules that actually inhibit the target of interest. In this case, we're looking for EGFR inhibitors. Then, out of the hits from your screen, so these are the molecules that inhibit EGFR exon 20 insertions, one needs to choose the lead compound scaffold for further medicinal chemistry to optimize the compound. And then lead optimization is the process in which all of the properties that are required of a clinical molecule are introduced into your compound of interest. And finally, if you're lucky, you end up with a clinical molecule. And there are many properties that are required of a clinical molecule. I've shown only 3 here on this slide. First, the molecule must be potent, so it needs to kill the tumor cells when administered to the patient. Then it needs to be selective, so equally important, it needs to not harm the normal cells of the body. And third, it needs to be stable. So this compound needs to hang around in the body long enough and be able to access the tumor in order to kill the tumor cells. So at first, when we saw, we were learning a lot of this as we were going along with our partners at Bayer Pharmaceuticals, and we had this image of this linear path resembling the air highway in Australia. But I have to admit that, in fact, our experience much more resembled the Passo San Baldo in Italy, so full of twists and turns and dark and foreboding tunnels. I'm not going to go through every single challenge that we encountered along the way, but just suffice it to say that we did encounter a challenge at almost every step of the process. But we persevered, and in the end, we identified our clinical molecule called sevabertinib. So there are many tests, many experiments that must be done before the FDA will agree to allow a candidate drug to be tested in clinical trials. Here I'm showing just one of these types of experiments that is performed to submit to the FDA. And in this experiment, tumor cells are taken from a lung cancer patient with EGFR exon 20 insertion mutations, and they're injected under the skin of a mouse, actually of several mice. And these mice are then treated either with inert vehicle or with varying concentrations of the compound of interest, in this case sevabertinib. And as you can see in the graph on the right, the vehicle-treated mice, shown in black, have tumors that continue to increase in size over time. On the flip side, the mice that were treated with sevabertinib had tumors that actually shrank. So this was a good result for the project.
And so we started by developing sevabertinib for the inhibition of EGFR, or epidermal growth factor receptor, which is a protein whose activity drives cell division. But it turns out EGFR is very closely related to a second protein known as ErbB2 or HER2, with very similar activity, also driving cancer growth. And you can see on the right side, exon 20 of the 2 proteins in the protein kinase domain. And you can see in the red letters and the pink letters in that diagram, you see numbers and letters. The numbers represent the position of what are called amino acids, the building blocks of the protein. And the letters are the codes. And you can see particularly in EGFR 764, 765, 766, and 767 says YVMA. And right under that for ErbB2, it also says YVMA.
Great. So almost as an afterthought, we decided to also test sevabertinib in models of HER2 exon 20 insertions. Now, you might have heard of HER2 also because it's a gene that's also involved in breast cancer. In breast cancer, it's actually usually amplified, which is one of those 3 types of genomic alterations that Matthew talked about at the beginning of the talk. In this case, in the case of breast cancer, it's typically not mutated, but just rather amplified to a very high copy number. But in this case, we have exon 20 insertions in lung cancer in this gene, just as we do in EGFR. So we took the tumor cells from a lung cancer patient, this time with an exon 20 insertion in HER2, injected them under the skin of several mice, and treated the mice either with the inert vehicle or with sevabertinib. And you can see, as before, as with the case of EGFR exon 20 insertions, here, the vehicle-treated mice had the HER2 exon 20 insertion tumors that continued to increase in size over time. But in the sevabertinib-treated mice, the tumors shrank. And together, of course, with our partners leading the clinical trials, our pharmaceutical partners with the associated physicians, sevabertinib entered phase 1 clinical trials. And here are the early results from these phase 1 trials that you're seeing here. In this graph, every single bar represents one patient. If the bars go up, the tumor in the patient is increasing in size, and if the bars go down, the tumor is decreasing in size. So you can see that many of the bars go down, but surprisingly for us, it was the bars marked in red, which are actually the HER2 exon 20 insertions, not the EGFR exon 20 insertions, that are responding best. So for us, this was a real paso san boldo moment. We had to immediately pivot and switch our attention. Instead of EGFR exon 20 insertions, now we were focused on HER2 exon 20 insertions. However, it was this data from the early data from the phase 1 clinical trial upon which the decision was based for the FDA to award sevabertinib breakthrough therapy designation. And this designation is a way to expedite the review process for candidate drugs that have the potential to provide substantial improvement over available therapy in areas of high unmet medical need.
And so this, sorry, go ahead.
I was just going to say that Matthew and I were arguing over who would get to present this slide, but Matthew will do the honors.
All right, so I think as Heidi said in that previous slide 2 slides ago, you have in the vertical axis the best percent change in tumor size, and in the horizontal axis you have patients. And this slide you can see is a little bit different results than the first exploratory study that Heidi showed, in that essentially almost every patient in this group, which is patients whose lung cancers have HER2 mutations within that kinase domain, whose structure that we showed earlier, and have been treated previously, but have not had previous therapies directed against HER2. Within this group of patients, all of the patients except for one had their cancer shrink, and 70% had what's called an objective partial response, which means you see that dashed line that's at 30% shrinkage. 70% of the patients had a tumor that shrank by at least 30%, which means really a good clinical response and a good clinical benefit. So then based on this result, on the next slide, the Food and Drug Administration, or FDA, granted what's called accelerated approval to sevabertinib for non-squamous, non-small cell lung cancers whose tumors have HER2 or ERB2 tyrosine kinase domain activating mutations. And then it also says who've received a prior systemic therapy. So this is called accelerated approval because it's based on this phase 1/2 study, which is a study that doesn't have a control arm. It's just compared to the historical experience. The full approval study is going to be based on what's called a phase 3 study, where you compare treatment with sevabertinib to the best possible current care that exists, and you see if it provides a benefit. And this is being done in patients with no prior therapy, again, based on these positive results. And then as you can see on the upper article, this is the first cancer drug based on science here from the Broad Institute. So it's been a remarkable road. We know that there are patients who are now being prescribed this drug, and we hear reports of benefit for these patients. And so it's really wonderful to have been able to take this road from scientific discovery to helping patients. And so what comes next, on the next slide, I mentioned that it's now approved for patients with previous treatment. There are ongoing trials for patients as a first treatment, which would be even more beneficial, hopefully. And then there are trials, Heidi mentioned earlier that HER2 alterations occur in other types of cancers, notably in breast cancer. And so this is another population in whom clinical trials can be considered. We mentioned how originally we developed this as an inhibitor of the EGFR kinase, and we can consider, our colleagues at Bayer are considering clinical trials for those populations. And finally, there's a growth factor called NRG1, which binds to and activates ERB2. And this is another oncogene. The gene for NRG1 is another oncogene in lung cancer, and the NRG1 fusion population is another population that can be considered for clinical trials with sevabertinib. And finally, there's so many people to thank. There's a long list that goes far beyond everybody we can put on this slide, but we especially want to mention Bethany Kaplan, who's sitting here and is going to join us on the panel, and who did some of the critical experiments that were on that long and winding road. And then our colleagues at Bayer Pharmaceuticals, including Franziska Siegel, whose picture is on the left, and Stefan Siegel, whose picture is on the right. Interesting coincidence. They are neither brother and sister nor husband and wife. They just happen to have the same last name. But it's become a very important name in cancer chemistry, in our opinion. And finally, just on the right side is an image of sevabertinib bound to the HER2 protein from our colleague here at the Broad, Benush Hajian, who's a structural biologist here in the Center for the Development of Therapeutics. So just thank you so much for all your attention, and I think Tom's going to lead a discussion coming forward.
Thank you.
Thank you so much, you two. And first off, I'm going to invite Bethany to come up on stage with us. Bethany was a research assistant working with Heidi and Matthew for 10 years? Yeah. Was that right? And did a lot of the basic laboratory work that led to the development of sevabertinib. So I want to start off just asking about the what you thought was the biggest challenge that you came across. I mean, you showed us the Pesasol and Baldo picture there with all the twisty paths and dark tunnels. Like, what was the darkest tunnel that you had to make your way through to get to this point?
So I think just Heidi may have a different opinion on this, and Bethany may have a different opinion too. But I'll start with my opinion, which is that the hardest thing was that protein structure studies of the exon 20 insertion mutants that had been done showed no difference between the cancer mutated protein and the normal protein. And all of the drugs which were different for the other mutants, they were more potent on the mutants than the normal protein. This wasn't true for the exon 20 insertion mutants. So the question was there some property of the exon 20 insertion mutants that made them impossible for making drugs? And people did big screens, and in fact, we did some big screens at the beginning of our project that still failed to find any difference. And so any molecules that could distinguish between the mutant form of the protein and the normal protein. So that overcoming that challenge to me was the biggest issue, and I think it was kind of a little bit of a leap of faith for us. So that would be my comment. But
maybe I'll go second, if that's okay. And so for me, I totally agree with Matthew. I also think a big challenge for this was the scarcity of appropriate cancer cell line models when we started the project. So there were that's zero cancer cell line models available when we first started. And cancer cell line models are important for two reasons. One, it's important for target validation. So if you remember, the first step in targeted drug discovery is showing that there's a dependency, that the tumor cells depend on this protein for survival. And the best way to do that is with a cancer cell that has the alteration that you're researching. So we couldn't do that at the beginning because we had no models. And the second thing is they're useful for evaluating compounds, and we didn't have these cancer cell models either. But what we did have was a cell model system that Bethany created, but we had problems along the way with that too.
Yes, we did. Yeah, as Heidi mentioned, we had to create our own system from scratch because there were no cell lines available. So I engineered a system where we basically artificially made a cell line that was dependent on the mutant form of EGFR or HER2, and used these to compare the effect of our compounds on the mutant form of the protein compared to wild type. There were a lot of technical challenges along the way. If you recall, one of them was the solubility of the compounds, which we didn't anticipate. It's like you just get thrown so many monkey wrenches throughout.
It's like, why is this not working? Is it the compound? Is it the cells? We don't know.
You'd be surprised just the number of things.
Yeah.
There were I ended up coming up with this system because of the solubility, where I had to use the pin tool, and then the issue where the cell titer glow wasn't lysing the cells properly, so I had to seal them all and flip them a bunch of times to get them to actually lyse the BAF3 cells, because they're these tiny little hematological cells that really just they were too robust to even lyse with the cell titer glow. So yeah, for me, it was all about those technical challenges that I just had to get through to get robust data when I was screening the compounds. But it was all worth it in the end.
So we could go on for 10 minutes about this, Tom, but probably you want to talk about something else.
Well, I do want to ask, Ashley, so Bethany, you had mentioned when we were emailing back and forth that there were lots of late night scouring data sets
yes.
before you actually realized that you had something. What was kind of like the first
Yes.
indication that you had that there was something promising?
Yeah, so it's funny, there was like I don't know, maybe 3 or 4 weeks there toward right before we were up against the clock, really, for a go, no-go decision with Bayer, because, you know, they're investing a lot of time and a lot of money in these projects, and if you reach a barrier that you can't get past, like Matthew was saying, you need to find a molecule that's going to be selective for your mutant over wild type, and he said, you don't even know if you're going to be able to do that. And once you've screened hundreds and hundreds and hundreds of compounds, like, at what point do you give up, right? So we had a bunch of compounds that had not yet been screened, and I was determined to get through all of them. I wanted them all to get through the assay before this date. And so I was up, at home. My husband's here, he'll tell you. I was sitting in the basement on gene data, analyzing data, and I remember very clearly that first compound that I saw, that was like, oh my gosh, we got one that actually has some selectivity for the mutant over the wild type. Like, we didn't know if we were gonna get there, and we finally got an inkling of hope that yes, we can do it.
An exciting moment.
It was a great moment.
Yeah.
And those aha moments are why we do science, right?
Exactly.
So one of the things that I was curious about, both in this presentation and just conversations we've had in the past, is the difference that you saw when you started testing things in patients, and you saw that cells that had the HER2 mutations were more responsive than the EGFR mutations. Like, why do you think that might be? This difference in response.
This is a tough question. I think I'll take it, unless you want to.
You could take it, and I might add a comment or two.
Okay, yeah. It's a tough question. So the compound was actually working really well against the EGFR exon 20 insertions in some of our cell models, not all of them actually. So this is interesting. So in retrospect, we're wondering if we should have paid more attention to the cell line models that it wasn't working as well in. But I think it's it's a little bit unclear as to why it's not working better for EGFR exon 20 insertion patients. Yeah.
Yeah, so my comment about this, after thinking about this question for years, is that you could pick a random person in this room or anybody else, and they'd have as good an answer to this question as I would. And so it's you have to look at what really happens, and it's striking, in drug discovery, as in many other fields, you can't always explain everything with what you know or what you can infer, and some things remain mysteries.
And that's also why the clinical trial is so important. There's only so much you can learn in preclinical models, like in petri dishes or even in mice. The clinical trial is the ultimate test of whether or not a candidate drug works.
So I have another comment, is that just to keep in mind, these patients weren't treatment naive. They all had received previous therapies, probably including the EGFR patients received EGFR therapies. The HER2 patients received HER2 therapies. We don't know what types of genetic alterations arose from those therapies and whether those would affect the ability of our EGFR compound to affect the EGFR patients, whereas maybe because the HER2 molecules are not, as a lot of them were antibody therapies, maybe they have different resistance mechanisms that didn't affect the ability of our molecule to affect those patients, whereas the EGFR molecules are also targeted therapies. Maybe their resistant mechanisms are also going to cause resistance to our molecule. So, there's a lot to consider.
Yep.
So that's why having those follow-on trials that you were discussing, Matthew, with hopefully first line treatment, that could teach us a lot. So I want to take a moment to sort of step back a little bit away from the specific sevabertinib project and just talk a little bit more about cancer drug development in general. We often hear that to develop a new drug from sort of basic discovery up until the moment of FDA approval, if it gets that far, can take 10, 15, 20 years. And I was wondering if you could talk a little bit about like why it takes that long. Like, what are the sort of like the lengthy steps in there? You showed us the sort of all the things that you're looking for when you're trying to decide on a potential molecule, but like, how long does it take for each one of those steps to sort of come to fruition?
I'll let you go on this one.
And I've asked Matthew this question before, and he had a heart attack.
So I'll make a few comments here. So really, our first discovery was Heidi's finding that where you saw those cancer cell colonies, they were either inhibited by drug or not, and she made that discovery, basically 21 years before the drug was approved for patient use, right? So the first step was really those, I think the first step is that discovery, and then, and it was published 1 year roughly after she made the discovery. But then, so the first step is that discovery. The second step is actually understanding if this is relevant in patients, right? And so Heidi made that discovery in cancer cell lines and in model cell lines, and then clinical data began to emerge over the next few years that said, yes, what we saw in cells is also what we see in patients. So I think the first few years was really understanding that or really that similarity between what we saw in cells and what we saw in the patients, that the existing drugs wouldn't work for this class of mutants. So then the second thing was kind of protein structural analysis, and people were making more and more EGFR and ERBB2 inhibitors and testing those inhibitors and saying, okay, these aren't working, and the protein structure is giving us information. So then the third step was really kind of taking this problem and getting somebody in the pharmaceutical industry interested in taking on the problem. And that step can be a really difficult step because, as Bethany said, it's a big investment. And I don't think if we really understood how big an investment it is, I'm not even sure we would have tried to persuade anybody, right? Because it really is, right? And so that's a big thing. So then once you get to the decision, okay, this is a target and we want to make a drug, that gets you to that winding road through the mountain passes that Heidi was talking about. And I got to say, Heidi was showing it in good weather, but in fact, I think you're trying to get through that in a snowstorm with 100-mile-an-hour winds, right?
Yes.
All right, and in a snowstorm and 100-mile-an-hour winds, and there's an earthquake happening at the same time. All right, if you really want to get to the degree of difficulty, right? Because so the first thing is actually to have an assay, right? So our first assay was like a biochemical assay. And so we found these compounds, okay, they're different between the mutant protein and the normal protein. That was a big breakthrough just to find it in the biochemical assay. But then, as Bethany says, we test them in the cell assay, and they don't work, all right? Okay, and so then there are all these decisions, like, do you keep going or do you quit? And this was a really, it's an easy decision for us. We thought we should keep going. but for the scientists at Bayer who were, investing heavily in this, that decision was more difficult. Right? And so then once you get a compound, right, getting a compound that works in a biochemical assay with pure protein, that's one thing. Then you have to get that compound. It has to get into a cell, which means the cell is surrounded by a membrane. It's a long answer, but there's a lot of stuff.
That's
all
right.
Just scratching the surface. The cell's surrounded by a membrane, and you have to get a compound that actually crosses that membrane from the outside to the inside, right, into the cell, okay? So that it can get to the protein.
It doesn't spit it out again.
Yeah, and it doesn't spit it out again.
Flux.
Yeah. And now you've got to get something that you can either inject into the blood or you can swallow as a pill, and it has to, this is the point Heidi made in her slide about it being stable, right?
Yeah.
So we talked about selective, right, is different between the cancer and the normal, and then it has to be stable. So you have to get it into the blood, or in this case, as a pill. So you have to be able to swallow it, and it has to get released into the blood, and it has to be stable and not broken down in the blood or in the liver or anywhere else in the body. And then it has to be potent, which means there has to be a small enough amount of it that it's not causing some sort of toxicity that's random, and also it has to be small enough that you could actually swallow it in a pill, right? Imagine like you have something that cures cancer, but the pill's this big, and you can't swallow it. All right,
Yeah.
so those are some steps, okay? And then you have to test it in animals, and you have to ask, is it toxic or not toxic? And so the first molecules, they worked in cells, right? They were potent. You give it in a pill to a mouse, but actually they had some problems, right? And so they had some toxicities, and our colleagues at Bayer were really pretty awesome with this. So you have to engineer the molecule and see, okay, can you have a molecule with all these good properties, but it overcomes the toxicities? All right. Now, all right.
And there's
We're
more.
on to
No.
chapter 4.
Right? And now, right, you're still not at the starting line of a clinical trial, right? So now you've got some data. You can say, okay, is it like it looks like it works in some models? Then you have to say, okay, does it actually work in a mouse with the disease or a model of the disease? And if it works in that mouse with a model of the disease, how toxic it is, right? And then with all those data, right, then the company can decide to go to a regulatory agency like the FDA and say, okay, here are our data. This is, again, what Heidi was saying to sort of prove those graphs that she showed where the tumor was shrinking. Okay, we can give this to patients. All right, so that's only getting it to patients, right? And so that's a really long and winding road. That took us from 2013 to 2021 going into patients, right? And then in clinical trials, again, as Heidi mentioned, there are a lot of unexpected surprises, including which patient population works. So long answer, scratching the surface of how many challenges there are.
Yeah.
And should also add, there were a lot of other molecules from other groups going into clinical trials. Most of those failed for one reason or another to solve the same problem. But there's one other one, zongertinib, that's also shown some success and also been approved by the FDA.
Which is great for patients, that there are two that now have accelerated approval.
So I'm going to ask one more question and then open up the floor for your questions. But so start thinking about what you want to ask. And like I said, you can flag down one of our mic runners. They'll bring a microphone to you. And again, if you have a really technical question, I'm going to ask you to hold it for the reception so that we can allow some of the community members to get their questions answered. So the last thing I want to, there's a little bit of a pivot. AI is everywhere now, right? It's seeping its way into everything. And as we've been talking about the challenges inherent in drug development, like, where do you see a role for artificial intelligence in this whole process? Do you think that there's ways in which it could make it simpler, make it harder? What are your thoughts?
I guess I inherited this question because Matthew took the last one.
I would try, but
Well, I'll say a few comments and you guys can add in. Yeah, I have to say I'm a little skeptical about use of AI in any way that's other than a tight partnership with a scientist who's actually doing all of these experiments and thinking through all the issues that need to be thought through. Each drug discovery project is very different. There's not a lot of, there's not so many similarities that you can just crank it through a single pipeline. If there were, then our project would look more like the air highway than the Paso San Baldo, and that's not what we experienced at all. One place where AI is really useful right now is in protein structures. So there are many tools, including AlphaFold, that can predict protein structures much better than we could, say, even 5 years ago. So that's a big advance for science in general. But I think the larger, contribution of AI, I have to say I'm personally a little skeptical that it's going to replace teams of scientists thinking about a problem anytime soon. But let's hear what my colleagues have to say.
I was going to say something similar about the structural biology and the helping with the modeling protein folding, but it does streamline data analysis quite a bit if you use it properly. I don't think it's going to help with the medicinal chemistry problems, which I also wanted to say Matthew really did only touch the surface of. I don't think you mentioned, I've been working at AstraZeneca for the last 7 years, so doing a lot of drug discovery, and I've been shocked at the number of roadblocks that you can reach through the process of drug discovery, just in terms of the chemistry of all of the things that you can encounter that the chemists need to overcome. And yeah, Matthew we could have gone on for probably
So
another
we
hour
have another
about
15 minutes.
Yeah. Well, let's talk about multi-parameter optimization then.
Yeah.
So yes, there's a reason it takes 20 years. But yes, I agree. I think in terms of the things that really need to be tweaked little by little as you encounter roadblock after roadblock, AI isn't going to do it for us. We're going to have to to do that ourselves. But there are a lot of ways that we can use it to streamline our analysis and our, the way that we have our workflows in the lab. So on my day-to-day, it definitely helps streamline things. But in big picture, it's not going to solve our problems.
All right, well, the floor is open to our guests in here. Somebody has a question that they'd like to ask your speakers? In the middle.
Thank you for your wonderful contributions and helping humanity. It's really amazing. The question I have is, there seems to be a debate in the cancer community between, a mutational approach to solving problems versus immune therapies like cancer vaccines and so on and so forth, right? Do you think, my question is, do both of them have their place, or do you think at some point, because of advances in cancer vaccine and immunotherapies, the latter approach would kind of dominate?
Do you want to take this one?
Sure. So yeah, I think it's a great question, the question again is, therapies target against really cancer growth pathways that are activated by mutations, as we've seen with the case of HER2 mutations in sevabertinib, versus immunotherapies, which may be responding to new antigens that stimulate the immune system. So for example, in the case of lung cancer, both of these approaches have been proving very fruitful. There are sets of patients who benefit from immune therapies that stimulate the immune system. These are mostly, in the case of lung cancer, these are mostly cancers that occur in smokers. And so these are cancers that have a large number of mutations. That means they have a large number of antigens. And so sort of stepping back a little bit, our immune systems are taught to distinguish our self from non-self. So that is to say, our immune system is really powerful, but it doesn't attack the normal cells of our body ordinarily, unless we get an autoimmune disease. But it attacks infections, viruses, bacteria, et cetera, that infect us. But it also means that if you make a mutation, it may be recognized by the immune system. So if you have a cancer with a lot of mutations, it can get recognized by the immune system. And that's why cancers that have a lot of mutations, like ones that arise in smokers, or melanoma in people who are ultraviolet light sensitive, these diseases tend to respond very well right now to immune therapies. In contrast, for example, the lung cancers of people who've never smoked, where we find the EGFR or the HER2 mutations, those cancers, in contrast, don't respond very well to current immune therapies, and they respond much better to these therapies that interrupt growth pathways. So we could imagine, for the foreseeable future, different approaches for different types of cancer, depending on their causative mechanism. And then we have to see, hopefully we'll be able to advance on all fronts in our fight against cancer. And perhaps one approach will become so successful that it will be used for all patients.
All right. We had somebody, the gentleman in the polka dot shirt there, I believe he had his hand up.
It's my friend William.
Yeah.
This is more of a general question. I have a son and his wife who have a baby coming along, and they keep putting out baby names. And I was just wondering, how do you name the baby sevabertinib?
All right. Well, first of all, William, congratulations on the impending birth. And in this case, we don't know so much, but I'm going to let Heidi answer this question.
So I have to say, because the two Bayer colleagues who spearheaded this project were Franzi Ziegel and Stefan Ziegel, we wanted to call the molecule Ziegelertinib, but that didn't fly. So actually, we academic scientists were not involved at all in the selection of the therapeutic name. That was done by Bayer's teams of lawyers and marketers, and so as it turns out, there are many rules which a small molecule name must adhere to. For example, it can't mean anything. It can't mean anything bad in any language. You just don't want it to do that. It can't say how great the molecule is, right? It can't be false advertising in any language. So there are actually rules about choosing the molecule name and then posting it to the WHO for, I don't know, whether it's 6 months or a year, but some length of time, so people can take a look at the name and comment on it. And there are certain letters it can't use because some languages don't have those letters. So it has to follow all of these rules. And then after the 6 month or 1 year, whatever it is, waiting period, then the name can actually be formally assigned. So I'm afraid that the answer might be the lawyers, but I'm not 100% sure.
Think about that the next time you're watching TV and you see a drug ad come on. Yes, up in the middle.
Hello. Thank you for the presentation. It was wonderful. I had a question about the workflow that you had and how you see this transferring. Now that you've taken sevabertinib through this process at Broad from discovery all the way into phase 1, 2, and hopefully soon 3, does any of that carry over to other drug discovery? Even though it's very different journeys, what can you learn now that you've driven this windy, dark, and stormy road?
I think the most important thing we've learned is actually the value of a really good pharmaceutical partner. I think that's the most important thing we've learned here. This is not something we could have done in isolation without our partners. The fact that we had such a good partnership where both sides of the partnership brought expertise to the table that complemented each other. I think, I would say the most important thing we've learned is how to design a good partnership, industrial partnership. I don't know if Matthew has a different comment.
Sure, so I'll make another comment on that partnership. I agree with Heidi totally. We couldn't have ever made it here without the partnership with Bayer. I think that from our side, I think we also brought a lot to this partnership, including a really very deep understanding of the biology of EGFR and ERBB2 from having studied these proteins for a long time, and a deep understanding of what the impact could be. I think it's very important to develop a kind of domain-specific knowledge, a feel for the molecules and the disease, which can be used to guide a view of what might be an effective drug. And I think that was something that we brought to this piece of it. And Bethany's actually really active in doing just this. So
I was going to say, I learned so much from working as part of this Broad Bayer collaboration, and then I moved over to working at AstraZeneca and took everything that I learned, from Heidi over the last 10 years that I was working with her, of course, but also that experience of learning how to develop a high throughput assay, how to make sure it's robust, how to make sure you have the proper controls so that you're looking at your selectivity in the right way. All of that is translatable to any project. How to, if you have no cell lines that are available, what models are suitable to engineer a cell line. Sometimes, we were lucky that in the system that we were using, which was a BAF3 cell, which are normally dependent on a particular growth factor for growth, we were able to put in the EGFR or exon 20 insertion mutation constructs, and then those cells could grow without the growth factor and then be dependent on the expression of the mutant protein in order to grow, making them dependent on it so that when we treat with compounds, it will actually kill the cells. Not every target is going to be able to work in a system like that. So you can't just be like, yeah, I'm going to stick my mutant protein into this system and it'll work. So recognizing when that's appropriate, when that's not appropriate, and obviously always concluding your proper controls and all of that. So that of course was translatable to any project. And then just persistence, lots of persistence.
And then I would imagine, Matthew and Heidi, your relationship with Dana-Farber, that also brings the patient experience in as well, right? I mean, I know you're not actively engaged in clinical care, but still you're in that environment.
Yeah, so just I thought about that. So I'm kind of part of the lung cancer group at Dana-Farber, although not caring for patients. And so that means that we spend a lot of time talking with our colleagues who are clinicians, going to their clinical conferences. And I do think this is one wonderful thing about the Broad is it's a very unusual scientific institution because it's a purely basic scientific institute, but it has partnerships with a number of hospitals throughout the Boston area, including the Dana-Farber Cancer Institute, the Massachusetts General Hospital, the Boston Children's Hospital, and beyond. And I think the Broad is pretty unique in terms of being a basic science institute that brings into itself this depth of clinical knowledge. And I think this was a really visionary feature of the Broad Institute. And I think we've all been very lucky to work in this environment and have this vision of improving human health. And so we're talking about the collaboration between us here at Broad and Bayer as a pharmaceutical company. But in a different way, this collaboration between Broad and the hospitals has been incredibly powerful.
I believe we had a question over here. 2 questions over there.
Hi, fantastic story. Thank you for sharing. So there are other undruggable mutations. So I wonder why what helped this exon 20 mutation became a successful story, and if there are any other ongoing projects or collaborations with the pharma today. Thank you.
Do you want to take that one, or?
Either way.
Yeah. Okay. Let me start, and then I think Bethany can talk also. Why don't you start?
Could you repeat the question? You were saying you were asking about other targets that are thought to be undruggable, and what the approach is for those.
Yeah.
More how this one became a successful story. What helped?
Oh, as opposed to other undruggable mutations or quote unquote undruggables.
I think that's a complex decision as to how one determines what target one works on. So it wasn't patient prevalence. So I think we haven't mentioned this yet, but this mutation occurs in about 2% of all lung cancer patients. So we're not, so it's not a high frequency mutation. But as targeted therapies become more and more targeted, it divides the patient population up more and more. And so it's actually not unusual now to have a targeted therapy that addresses a small patient population who has the exact mutation that the targeted therapy is designed against. But why this one and not others? I mean,
why was it successful?
Yeah.
I think sometimes you get lucky. I'll be honest. I would say, like, currently at AstraZeneca, we're working on a lot of targets that are thought to be undruggable, and sometimes you at least give it a try, right? You give it a screen, you try the screening approach, you try multiple screening approaches, and sometimes you don't find anything, or you see a paper where somebody, they actually were able to get something that bound to that domain. Nobody's ever targeted that domain before, so you might develop an allosteric inhibitor or something that's preventing a protein-protein interaction as opposed to something in your enzymatic domain. So I think we're taking a really varied approach to what we're considering druggable nowadays, at least at AstraZeneca. And yeah, sometimes you get lucky and sometimes you don't. In this case, I think we always thought it was druggable.
We were never concerned about that.
We always thought it was druggable, right? It's just a matter of getting that selectivity and not hitting the wild type. And it's just, you just have to get the right molecule. And we've got there.
So why did we think this was druggable?
No.
And where does that fit in history? So we thought it was druggable because this is an enzyme, so it's a protein that catalyzes a chemical reaction. Catalyze means it makes it easier for a chemical reaction to occur. So when you have an enzyme, you have an active site, which is a particular structure of a protein, and you can fit something into it. And in this case, right, what you normally fit into the active site of a protein kinase is the substrate, the protein that it modifies, and adenosine triphosphate, or ATP, which gives a phosphate group that takes part in the reaction, right? So you've got this active site. That being, and we knew already, I mentioned right before we started, Bill Sowers and I started on protein kinase sequencing, we already knew there was this drug, imatinib or Gleevec. It was binding to the active site of a protein kinase and working. But if you go back, and that was 25 years ago, but if you go back another 10 years before Gleevec, it was thought that protein kinases as a whole class of proteins were going to be completely undruggable because that active site is pretty highly conserved and they all bind the same substrate, ATP, and it was highly conserved, and people said, you'll never get selectivity between protein kinases. And so that was that trial and error process that Bethany described that got there. If we think about types of proteins that are considered undruggable today, I'll just give an example. A lot of transcription factors, proteins that bind to DNA and they tell the DNA to make messenger RNA, a lot of those are considered not to be sufficiently specific. Specific is the word that I was trying to say before getting tongue-tied. They're thought to be not sufficiently specific, and you can't make a drug until you find some trial and error optimization to get there. And so it may be simply that willingness to keep trying and then let people try different approaches, right? One interesting thing about sevabertinib, so there were other EGFR and ERBB2 inhibitors out there. The molecular structure of sevabertinib is completely different. We were not reasoning by analogy to previous structures. And people had tried that also for the exon 20 insertion mutants, and that failed. So we took an open-ended approach.
If I can add one thing.
Yeah.
It was also thought that at the time that we started this, that it was not known whether we would get specificity for the EGFR exon 20 insertion over the wild type EGFR. It wasn't known if we could get mutant specificity with a small molecule. So that was, that was kind of a risk that we took when we started the project. And it wasn't until we started developing chemical matter that we realized we could get selectivity for the mutant versus the wild type.
I think we'll take one more question over here.
Thank you for the talk today.
There's also one guy over there.
So my question is, can you say more about how to set up a successful partnership and any kind of like governance structures that worked really well for both the Institute and Bayer? I know it's been many years in the making, so like anything that you remember working really well, I would love to learn more.
Yeah, I think that the most important thing is the project teams that were comprised of scientists both from the Broad and from Bayer. So I had a counterpart project team leader that was Franzi Ziegel at Bayer, and the two of us worked together on the biology side to propel this project forward. We had many face-to-face meetings of the entire project team and the leadership of the collaboration. So I think this equal both having skin in the game, this equal commitment to the project is important. And I think that was the important way that the collaboration was set up. I don't know if Matthew has any more comments.
Sure, I'll just add one more thing. I think that was really great about this partnership is we actually had the same goal in mind, that we actually wanted to make new drugs that would treat cancer patients. And a lot of partnerships, people don't actually have the same goals, right? Their goals may be a little bit at right angles, and I think that was super important.
Yeah.
Well, if I can beg your patience, we can take one more question over here.
Yeah.
I heard you say that the HER2 mutation is not really correlated with tobacco use.
I'm sorry, we're getting a cough at the same time. Can you say it once
Speak
more?
up a little bit.
I heard you say, I thought I heard you say that the HER2 mutation is not correlated with tobacco use.
Correct.
And my question is, you also mentioned something about 2%, and the underlying thing is that these days tobacco use is down, but marijuana use is up. Have you seen any correlation with lung cancer in general with marijuana use? So two-part question, so to say.
It's a great question. I don't think we've seen anything, but I'm not sure we've looked carefully enough. So that's an interesting question.
I'll say a couple. I'm going to answer that two ways. First of all, the marijuana use, I don't think we have good enough data on large populations of people using marijuana for enough time to evaluate the impact of marijuana smoking on lung cancer. And I'm not as familiar with the patterns of marijuana smoking as I am with tobacco smoking. Many people who smoke tobacco will smoke 20 or 30 or 40 cigarettes a day. I believe that most people who smoke marijuana do so significantly less frequently than that, and therefore the dosage may be less. But the second thing that we also don't know, right? So many people who use nicotine products now do electronic cigarettes or vaping rather than tobacco smoking, and I don't think we know what's the disease impact of vaping, and will that be safer in terms of cancer, or the same or less safe than tobacco smoking. And I think as we have an increasing population of people who are vaping, it will be very interesting to understand what that effect is. And I think those studies are beginning to be underway, but it's a really important question.
And there are always more questions to ask. And with that, thank you, Matthew. Thank you, Heidi. Thank you, Bethany. And thank you to all of you for joining us this evening. If you're in the auditorium, please join us for the reception in the Broad Discovery Center. And if you happen to be looking at YouTube, please mash that subscribe button on the Broad channel. And thank you again for joining us tonight.